Talk about codominance. Some genes have alleles that are both expressed, and there are three phenotypes. There is not a recessive allele, and both are dominant. When we write out the genotypes for codominant problems, we only use a capital letter, and we use superscript for the two different versions. So for example, if the trait is color for C, the different versions would be if it's blue or white color, or blue and white. So we use lowercase b's or capital B's to represent superscripted to represent that color. The specific color is blue. And then we use W here to represent white, and then BW represents blue-white. Codominant alleles have three phenotypes. Sickle cell anemia is a genetic disease, and that's an example of a codominant disease. It affects the hemoglobin of the red blood cells. The hemoglobin is normally a ball-shaped molecule, but when the sickle cell allele is there or present, it forms a long strand. And then when the red blood cell carrying the molecule distorts it into characteristically long shape. So here's a blood smear, and the top left one is our normal one. These larger cells right here are white blood cells, and the smaller ones here, these are all the normal characteristic of red blood cells. So on the second image on the right is the sickle cell anemia. We do see some normal cells like here and here. So not all the cells in this case are sickled, but then we also see the sickled cells, which are these elongated cells right here and here. The genetics of sickle cell anemia. The shape of the hemoglobin molecule is controlled by two alleles. We have this normal allele, and then we have the sickled cell allele. There are three phenotypes that are present when with the different variations of the allele combinations. We have a normal. These individuals have two normal hemoglobin alleles. Or we have the sickle cell anemia. That's a severe form where all the red blood cells are affected. Sickle cell anemia patients have two sickle cell alleles in their genotype. Or you can be a heterozygous condition where we have a sickle cell trait, which is a mild condition where 50% of the red blood cells are affected. Sickle cell trait individuals are heterozygous, and they have one of each allele. We already talked about using the capital case and the superscript to identify the alleles. So a normal hemoglobin we'd identify as H superscript N. Sickled hemoglobin is H superscript S. When we have someone who has normal hemoglobin, they would have the two H's, two superscript N's. If someone has the trait, that would give us the N and S, and that's where we have 50% are sickled. And if someone has the two S's, alleles, 100% are sickled. So here's an example of a cross between two individuals. We have parent 1 who has the sickle cell trait, which is that they're heterozygous, and it's crossed with parent 2 who also is heterozygous. In the end, you're going to have a 1/4 chance or 25% chance that it will have no sickle cell or the trait or anemia. So this would be normal. We would have a 50% chance, or 2 out of 4, to have the sickled trait, which is 50% sickled, or we can have the 25% chance of having 100% sickled. So a 1 in 4 chance of having a normal child, a 50% chance of having a child who has a sickled trait or 50% sickled, and a 25% chance that you would have a child with the anemia, which means all of their blood cells are sickled.